Water loss from leaves is an inevitable consequence of photosynthesis. To obtain carbon dioxide, stomata must be open to the atmosphere. Having open stomata also opens the leaf to evaporative water loss. Adaptations to dry conditions involve decoupling water vapor loss from CO2 uptake. Plants do this through employing different types of photosynthesis. Among the green plants, there are three broad photosynthetic types. Under normal conditions of ample water availability, green plants engage in so-called C3 photosynthesis, which is the default form, if you will. Under arid conditions, many plants engage in so-called C4 photosynthesis. This type of photosynthesis is widely found in plants that inhabit arid conditions, including the grasses, sedges, and euphorbs. Finally, there are many arid-adapted plants that use so-called crassulacean acid metabolism, or CAM photosynthesis. CAM photosynthesis is also widespread among desert-adapted plants, including the cacti, many of the southern African succulents, and the agaves and aloes. CAM photosynthesis is found among the most drought-tolerant plants. Let's first give a broad overview of photosynthesis in green plants. All photosynthesis is a two-step process. First, there's a light reaction where energy in light is used to strip a proton, that is, a hydrogen ion, from water. This is the step where oxygen is produced as a waste product. This is very important because oxygen is only produced under light conditions. The protons, which are very energetic, are then channeled to the dark reaction, where they are combined with carbon dioxide to produce carbohydrates. The dark reaction usually ends up with ATP being used to reset the dark reaction. Let's turn now to normal, or C3 photosynthesis. Here's the cell contained within the open air spaces of the spongy mesophyll, which opens to the outside air through a stomatum, bordered by two guard cells. The dark reaction of C3 photosynthesis takes place within the cell and revolves around a molecule, ribulose bisphosphate carboxylase, or Rubisco for short. CO2 is fed into Rubisco, which forms two molecules of 3-phosphoglycerate, or 3-PGA for short. These both contain 3 carbons, which is where C3 photosynthesis gets its name. One of these PGAs cycles back to reform Rubisco, consuming energy in the process. The other feeds into the production of carbohydrate. This is carbon fixation. C3 photosynthesis sets up a conflict between water vapor loss and oxygen loss through the stomata. When the stomata are open, oxygen produced by the light reaction diffuses out of the cell into the air spaces of the spongy mesophyll, then out through the stomata to the air. Water vapor leaves the leaf through the same open stomata. Thus, water vapor loss and oxygen loss are coupled. A conflict arises when water becomes scarce. The plant conserves water in the leaf by closing the stomata. This keeps water in the leaf all right, but it also keeps oxygen in there, elevating oxygen concentration in the leaf's air spaces and in the cells. Here's where the conflict arises. Oxygen competes with CO2 to bind with Rubisco. Elevated oxygen within the cell binds to Rubisco, excluding CO2, a process called photorespiration. Now, Rubisco does not break down into two C3 molecules, but one 3-PGA, which feeds back to regenerate Rubisco, and a 2-carbon molecule, 2-phosphoglycolate, or 2-PGC for short. 2-PGC cannot feed back into carbohydrate production, and this eliminates the leaf's ability to fix carbon into carbohydrate. This is why C3 plants generally do not do well under arid conditions. Their principal means of water conservation, closing the stomata, also blocks carbon fixation through photorespiration. C4 photosynthesis is one way plants avoid this dilemma. C4 photosynthesis uses a different enzyme to capture carbon, this one called PEPCO, or phosphoenol pyruvate carboxylase, as opposed to Rubisco. Instead of CO2 being captured as 3-PGA, a 3-carbon compound that feeds directly into carbohydrate synthesis, CO2 is now captured in the form of oxaloacetate, a 4-carbon compound. This is where C4 photosynthesis gets its name. There are 2 advantages to capturing CO2 into oxaloacetate rather than 3-PGA, as C3 photosynthesis does. First, oxaloacetate can be stored for use later. It needn't be used right away, as 3-PGA must. The second is that PEPCO is far less susceptible to photorespiration than rubisco. This allows the C4 plant to decouple water vapor loss from carbon fixation. During the day, when light energy is being captured and oxygen is being produced, the plant can close its stomata, blocking water vapor loss and oxygen loss. The increased oxygen concentration in the cell does not block carbon fixation, as it does in C3 plants, because PEPCO is not susceptible to photorespiration as rubisco is. Carbon fixation, therefore, proceeds apace, storing the fixed carbon into oxaloacetate. At night, when the light reaction is no longer generating oxygen, the stomata can open, allowing the built-up oxygen to leave the leaf. Water vapor loss is now lessened because external temperatures are cool and humidities are high. Oxaloacetate is then mobilized and fed into the rubisco pathway for conversion to carbohydrate. Because oxygen concentration within the cell has dropped, photorespiration is no longer a problem. Carbon fixation is therefore decoupled from water vapor loss in C4 plants. CAM photosynthesis provides an additional means of decoupling carbon fixation from water vapor loss. Like in C4 plants, CAM photosynthesis provides a workaround to rubisco's susceptibility to photorespiration from high oxygen in the leaf during the day. At night, the stomata of CAM plants are open. Water loss rates are low because the leaf is cool. Now, CO2 is able to enter the spongy mesophyll from the atmosphere, where PEPCO converts it to oxaloacetate, as in C4 photosynthesis. Rather than leaving the carbon stored as oxaloacetate, vacuoles within the cell convert oxaloacetate to another 4-carbon storage compound, malate. Later, when the sun rises, the stomata close, conserving water. Energy from the light reaction then converts the stored malate directly to carbohydrate, bypassing rubisco. Again, fixation of carbon in sugars is effectively decoupled from evaporative water loss.